Different antibiotics have different modes of action. This is because of their structures and degrees of affinity to certain target areas within bacterial cells. For example, antibiotics can inhibit protein or nucleic acid synthesis, or damage or disrupt the cell membrane. Alternatively, antibiotics like penicillin work by inhibiting the synthesis of the cell wall. The cells of humans and animals do not have cell walls, but this structure is critical for the life and survival of bacteria.
The modes of action of both early sulfonamides and modern penicillins revolve around the concept of enzyme inhibition. Most enzymes will bind to a specific molecule called a substrate. The binding site on the substrate molecule must be complementary to the active site of the enzyme. Sulfonamides act to inhibit the synthesis of DNA within bacteria. This does not kill bacteria outright, but it inhibits the growth and multiplication of bacteria, giving the body's immune system time to fight the infection. As such, sulfonamides are called bacteriostatic agents, which inhibit growth of bacterial colonies. Penicillins and other beta-lactam antibiotics are bactericidal, as they actively kill bacterial cells. One important sulfonamide is prontosil, a red azo dye, which was found to be antibacterially effective in vivo, but ineffective in vitro. This arises because prontosil is converted into the active sulfonamide by bacteria present in the small intestine of mammals. Sulfanilamide is an effective antibiotic due to its similar shape to para-amino benzoic acid, or PABA. PABA is required for the production of tetrahydrofolate, which is needed for the synthesis of DNA in bacteria. The conversion of PABA into tetrahydrofolate is achieved by the enzyme DHPS, which binds to the PABA molecule at its active site. As the structure of sulfanilamide is so similar to that of PABA, the DHPS enzyme is not able to differentiate between the two. Both have complementary shapes to the active site in DHPS. So sulfanilamide can occupy the DHPS active site, preventing para from binding to DHPS and therefore preventing the production of bacterial tetrahydrofolate and DNA. Although discovered later on, penicillins have proved to be the preferred method of treatment. Their action leads directly to cell death, rather than just the containment strategy provided by sulfonamides. Unlike animal cells, bacteria possess cell walls. For gram-positive bacteria, this is comprised of a thick peptidoglycan wall. In contrast, a thinner peptidoglycan layer, supported by an outer liposaccharide membrane, encapsulates gram-negative bacteria. Individual peptidoglycan molecules are long-chain polymers made by linking together sugars and amino acids. When linked together, many peptidoglycan chains can form a rigid wall which protects the bacteria from harmful chemicals and stops them swelling and bursting. The peptidoglycan wall is constantly being assembled and repaired. This is accomplished using a transpeptidase enzyme, which is able to link amino acids at the end of two separate peptidoglycan chains, joining them together to form the strong peptidoglycan structure. The mechanism involves two nucleophilic acyl substitution reactions. The first reaction forms an intermediate ester, which reacts to form a new amide bond that links the peptidoglycan chains. Penicillin acts by inhibiting the transpeptidase enzyme. It mimics a peptidoglycan chain, and an ester is formed that joins the penicillin to the enzyme. As the penicillin group is so large, it prevents attack of a nucleophile at the ester carbonyl, and so the ester does not react with the second peptidoglycan chain. With the enzyme unable to form crosslinks, the peptidoglycan wall begins to degrade. No new peptidoglycan chains can be added to the cell wall, and eventually the cell bursts.